Human chimeras, the future of organ transplants. The term chimera comes from the lion-snake-goat hybrid monster in Greek mythology, but in a biological context, a chimera or chimerism refers to an organism that is a combination of two or more genetically different individuals. You might be thinking, so what? Well, with over 100,000 people in the US currently in line waiting for an organ transplant, further development of chimera technology can be used to engineer animals to grow human organs. This would provide us with an unlimited supply of organs needed for transplants as well as reduce the risk of rejection. The methodology of creating human-animal chimeras integrates stem cell technology and genetic modification. So, let's break it down. What are stem cells? Think of stem cells as cells that are looking for employment, and through the process of cell differentiation, these stem cells are hired and become specialized. Once hired, these cells have a specific function to carry out in the body. There are two main classifications for stem cells, embryonic and adult. Embryonic stem cells tend to be more focused on as they are pluripotent, meaning that they have the ability to differentiate into most types of cells. But how do we get from there to a full-blown animal hybrid? In 2010, Japanese scientist Hiromitsu Nakauchi published how he was able to grow a rat pancreas inside a mouse using blastocyst complementation. Geeky, right? But a blastocyst is simply an early-stage embryo, a ball of cells that scientists can re-engineer. So, how does it work? Firstly, scientists take a rat blastocyst and delete a gene called PDX1 from the rat DNA, without which the rat is unable to produce a pancreas. The scientists then extract mouse pluripotent stem cells and inject them into the developing rat blastocyst. This is where the pluripotent stem cells mentioned earlier come into play. The mouse cells which have the PDX1 gene can fill in the missing DNA of the rat embryo. The mouse pluripotent stem cells would differentiate and grow into a pancreas. The blastocyst now has a complete genome and can be implanted to birth a healthy rat chimera pup. However, this paper is only a proof of concept. It is important to note that chimerism with humans is much more difficult due to many factors, such as evolutionary distance and ethical issues. But for now, chimerism is a fascinating occurrence in biology. With further scientific development, it may just be the future for medical transplantations. Who knows, someday maybe even you could have a pig-grown stomach.